Okay, so chapter 5, chapter 4 is going over the different parts of the cell. Chapter 5 is really getting into the cell membrane and how exactly do we do transport in the cell? How do we bring things in? Let me adjust the, yeah, that works a little better, there we go. Okay, how do cells signal with each other? How do we bring things in? And then what is the structure of the cell membrane? So, so just getting an appreciation of why we need a cell membrane. So we need to have some way of separating the outside of the cell from the inside of the cell, right? Because if you couldn't separate the outside from the inside, think of, when it's wintertime, if you didn't have a way to if the outside became the inside of your house, you would probably freeze to death or something, or you'd at least have to walk around with a lot of blankets. It depends on how cold it is wherever you're at. So similar thing with the cell. We want to, if you drink some coffee or you eat a bunch of Sour Patch Kids, you don't want something that's super acidic to go directly into all of your cells. You want to have some way of only bringing in what you need from your bloodstream or outside of your cells in the extracellular matrix, only bringing in what you need, and you want to have some way of getting waste outside of your cell. So anyways, that's what we mean by selective permeability. We want to be selective or choosy about what we bring in and what we choose to leave our cells. And then permeable is just this idea of things leaving the cell. Okay, so this is just showing two different nerve cells communicating with each other. There isn't really a ton to mention here. It's just, I don't know, it's just kind of there. I thought it was sort of a cool picture. All right, 5.1. So this is really getting into the actual structure of the cell membrane. We call it a fluid mosaic model of lipids and proteins. Let me just really teach from this picture. So fluid, because all of these components are actually very fluid, meaning the main part of the cell membrane is, we call it a phospholipid bilayer, right? Phospholipid bilayer. And this phospholipid bilayer, if you removed all the proteins, all the other stuff from this image, and you just had the phospholipids, which is, this little head and that tail, that was all that was part of the cell membrane, that would actually be super fluid, almost like a soap bubble, a lot of movement, because it's really just kind of fat, sort of not completely solid fat, but a little more, it's really more like olive oil without these other components in it. Anyways, so that's what they mean by fluid. Mosaic comes from especially these different proteins that are all scattered throughout the cell membrane. And then you include the extracellular matrix and some of the cytoskeleton. It looks like a mosaic picture, I guess. I'm not really an artist, so I kind of have to take their word for it. But there you go. So let's get into some of the parts of this picture. I do want to talk about the, so if I draw the phospholipid, so if I were to zoom in right on one of these phospholipids, we say the phospholipid is amphipathic. So amphipathic, I think I spelled that right. Let me go back a slide. Yeah, amphipathic. Where what that means is there is a hydrophilic part. So that's the hydrophilic head. And by hydrophilic, we mean polar. So this head wants to be, you have the hydrophilic head here, hydrophilic head here. It's a phospholipid bilayer, 2 layers of phospholipids. These heads want to be, they're hydrophilic, meaning their hydro is water, philic is loving. They are surrounded by water because on the outside of your cells and on the inside, they're surrounded by water. So that's one part of it. And then the tails are hydrophobic. So I don't know, amphipathic, I don't know, I think it's something like 2 parts. I don't know, maybe an amphitheater, it's kind of like I have no idea where I'm going with that. It's just got the 2 parts to it. Hydrophobic and nonpolar go together. So because these tails are made of a bunch of nonpolar bonds, they behave hydrophobically. Water's a polar molecule, right? It's got that H2O, it's got that partial negative charge. What even is that? It's got that partial negative end with the oxygen and a partial positive with the hydrogen. It's polar, two ends, negative, positive. Hydrophobic is the opposite. These tails don't have the positive, negative charge to them. So that's why they're on the interior. So the interior here, where you have the two different tail layers coming together, that's going to be secluding water because it's made of all those non-polar bonds. Okay, and then these things to be your proteins, where these are integral proteins. They're integral because they span the cell membrane, and then these smaller proteins there, and then really just there. These are called peripheral proteins. So if you're on the periphery of a room, that means you're on the outside. So they're peripheral proteins because they're the room being the cell membrane. They're on the outer portion of the cell membrane. They're not spanning the cell membrane like the integral proteins. Okay. And then this right here, I'll do this one. This is cholesterol. And cholesterol, if I draw it a little bit bigger, it has those hexagon shaped things. A big old string of them. What even is happening right now? Anyways, those are steroid molecules. Remember I said that those look like you're at the gym. Looks like little weight plates. So cholesterol, it's a steroid molecule. Those are scattered throughout the cell membrane. And why am I writing like that? Let me write like a normal person. So cholesterol. And this is interesting. Cholesterol is very interesting. So if you're at high temperatures, so if the cell's in an environment where it's high temperature, it will make so when something's at a high temperature, if you're the temperature's really hot right now, at the molecular level, that means the molecules are moving around faster, right? So if a cell is if your body temperature gets to 102 or something, where it's elevated, the molecules that make your cell membrane, they're moving around faster. And the reason why it's a bad thing is if these things are moving around faster, that means more gaps, more spaces can open up between those phospholipids and let certain things in and let certain things out. Well, we said earlier that the cell membrane is selectively permeable, meaning we don't want to disrupt that balance of letting things in and out. And so at high temperatures, you start letting too many things in and too many things out. Well, what cholesterol does is it's going to prevent, or prevent wouldn't be the right word, it makes the cell membrane less fluid. Meaning they prevent, the cholesterol will kind of act almost like putting these cones or like a roadblock or something. It prevents the traffic. If each of these phospholipids is like a car, it prevents them from moving around too fast. So you don't get as much space between each of the phospholipids, which means you don't have a bunch of stuff coming in and going out that you don't want to go in and go out. Now the opposite happens if it's a really low temperature. It's really cold outside. Think of if you ever made bacon or something, and you have that bacon grease that's made of lipids, made of fat. As it cools down, it starts to solidify, it starts to harden. So the opposite thing happens. At low temperatures, these phospholipids start to cluster together and they start to clump together. And you don't want them to be too close together. You don't want these cell membranes need to be fluid because they do need to let things in and let things out. But if they get too clumped together, that disrupts that balance. And so at low temperatures, the cholesterol will make the cell membranes more fluid. So in other words, the cholesterol acts like a buffer. Think of the cholesterol like a buffer. If it's a really low temperature, it's going to make the membranes more fluid. If it's a higher temperature, it makes them not as fluid. Really important idea. So, for example, if you're a fish that lives in like a super cold environment, you probably are going to have, or you're more likely to have, more cholesterol in your cell membranes, so that you can prevent the lipids that make up your cell membranes from getting too close together and freezing. Okay, yeah, a lot going on with this picture. These are the collagen fibers. So, this is part of the extracellular matrix we went over last class. So the collagen fibers really give that extracellular matrix some strength. The glycoproteins, so these carbohydrates here, those carbohydrates are really helpful in signaling. They act like a fingerprint for the cell. So meaning how like our cells can know, like, oh, that cell belongs in this tissue layer, like maybe that cell belongs in our body, or maybe that's like a foreign invader cell. Or even better yet, blood typing, like type A, type B, type O blood. You're like, well, what makes something have different kinds of blood types? Well, a lot of that comes from these carbohydrates that make up, so proteins that have those carbohydrates, those are called glycoproteins. So the glyco in glycoproteins is referring to the carbohydrates. So these proteins will have these carbohydrate fingerprints, like your fingerprints, my fingerprints are different from your fingerprints. That allows these cells to look different from one another. Okay, so it's a really important role in cell communication. All right, let me see if I got everything here. Oh, there's also glycolipids. And they would sort of have a somewhat similar role to glycoproteins, but the lipids would be attached directly to the lipid in the phospho, the lipid in the phospholipid bilayer. Notice the microfilaments here. These are the microfilaments. Microfilaments are made of the actin. I don't need to write that down. Okay, so yeah, just trying to bring together a lot of different kinds of information. Amphipathic, hydrophilic, hydrophobic. Yeah, cool. So, oh yeah, okay, this is kind of an interesting idea. So if I'm a protein, like going back to this picture, like I'm an integral protein like this. If I'm an integral protein, like this protein here, this portion of the protein right here, that's inside the cell membrane, this is going to be made of nonpolar amino acids, because nonpolar amino acids need to be able to interact with other nonpolar molecules, these phospholipid tails. Whereas this portion of the protein, these are going to be made up of so bad. This is I've literally never written with this marker. Polar amino acids. I don't know why I made it. There we go. There we go. Those are made of polar amino acids. They're polar because this section of the protein is interacting with water. Okay. Groups of certain proteins or lipids may associate in long-lasting specialized patches. Meaning these proteins, they're not just randomly scattered throughout the cell membrane. They actually will have, like, you might have one part of the cell membrane that specializes in, say, I don't know, bringing in glucose or something. Or like one part of the protein that specializes in transporting different ions across the cell. So I think the idea there is efficiency. That's the idea of like an assembly line. Like each person in an assembly line has a specialized job, like their own role. And it would be a similar principle here, where if you have each, like, different parts of the membrane specialize in a different process, it will just make the cell overall be more efficient. The fluidity of the membrane. So these phospholipids, they're not stationary. Like they're actually constantly moving around, meaning like they can swap positions. Like these phospholipids could switch positions with each other, and they could switch positions. They're constantly moving, where overall the whole structure, the whole cell membrane is solid, like it exists, but it exists with like constant turnover. They're constantly switching spots. And same with the proteins. The proteins they don't move as much as the lipids that make up the cell membrane, but they're also technically like shifting spots and moving around. And the movement of the proteins, some is more just random. Others can be in a directed manner where, as I said, we want to have those specialized patches of proteins that do a certain job. Others don't move around as much. They're more anchored to the cytoskeleton or the extracellular matrix. So a little random, but say you're skeptical. You're like, how do we know these membrane proteins move around? How do we even test that? Well, actually, back in the 70s, they did this experiment where they took a mouse cell, where they have the proteins on the surface of the mouse cell in pink here, and then we take a human cell whose proteins are represented with the blue color. And they were able to make somehow this hybrid cell. I don't know how functional this hybrid cell was. I'm sure it probably broke down after a certain period of time. But anyways, they're able to get to survive for long enough where they could see a mixing of proteins after 1 hour. Meaning, if proteins didn't move around in the cell membrane, what you would expect when you make the hybrid cell is the pink cells would stay here and the blue cells would stay the pink proteins would stay here and the blue proteins would stay there. But after 1 hour, you actually get a mixture of them. They actually just kind of all mix in together. I think of girl, boy, or something, or all the girls over here, all the boys over here, and then this would be them acting normal and they just mixing together. So there you go. Unit 1, you should have heard of this, gone over this a little bit with the unsaturated versus saturated fatty acids. So where there was a picture showing olive oil, your fats that are liquids at room temperature, they have the unsaturated fats, where the unsaturated comes from the double bonds. So in these fatty acid tails, it's this long chain of carbon atoms, carbon and hydrogens. This unsaturated, you would have some double bonds, where these double bonds cause this kink, this tail, instead of without the double bonds, if it's like our saturated fats, where it's just a normal carbon chain without double bonds, it's very straight, you can pack them together very easily, but these kinks that come from the double bond prevent us from packing as many phospholipids together in the same volume, the same area. And on that's what's happening at the molecular level, but big picture wise, this would cause this to be a liquid at room temperature, whereas these ones would be more like a solid at room temperature. So this would be actual butter, a stick of butter would be made of a bunch of saturated fats, whereas olive oil or something is going to have a lot more unsaturated fats, or vegetable oil or something. And then I mentioned this already on the fluid mosaic model picture, where cholesterol will do that moderating role, preventing the phospholipids from getting too packed together or from being too far apart and too fluid. Viscosity, that's if you're really viscous, it's a measure of how resistant you are to motion. So if you're really viscous, that means you don't move very well. If you're not very viscous, so this is more viscous, this is less viscous. So it's the opposite of fluidity. Okay. Mention this first bullet point. I mentioned this with going over the whole cholesterol thing. As temperature lowers, things solidify. Now, the temperature that things solidify is going to depend on what kind of lipids will make that up. Meaning, if you have a cell membrane that has more unsaturated fats, so if you had, this is going back to a fish that lives in a really cold environment, their cell membranes not only might have more cholesterol, but they would especially have more unsaturated fats, because those unsaturated fats can prevent that membrane, that packing from happening, and so it's going to keep their cell membranes from freezing. Or at least the temperature would have to get a lot lower for their cell membranes to freeze together. This is just in words explaining how cholesterol kind of has that buffer role with temperature and membrane fluidity. Yeah, not all species are going to have, or even within a species, not every person, not every species is going to have the same composition of lipids. It's just going to vary. Basically, the cell membranes of different species will evolve to adapt to their environment. Whatever kind of combination of phospholipids you're going to need to better survive in that environment. Oh, in some organisms will have the ability to change their lipid composition. So maybe if it's an environment where the climate changes a lot, it might be beneficial to have the ability to change maybe the amount of like saturated versus unsaturated fats. So maybe if it's a colder time of year, you'll start adding in more unsaturated fat lipids to keep the membranes from packing together. Where if it's warmer outside, maybe you have more saturated fats, or just less unsaturated. Maybe you start adding in more cholesterol or something like that. I don't know. I'm kind of making that up, but okay. Okay, so yeah, getting into some of these different membrane proteins. I mentioned like the fluid mosaic model. The mosaic is referring to the different proteins that make up the cell membrane. There's a bunch of different functions to these membrane proteins. And there were the integral proteins and the peripheral proteins that I referred to, where the integral proteins, this is an integral protein, you had like the hydrophilic parts, and these are the hydrophobic parts. The region that, this is called the transmembrane region. So that's another word for like the hydrophobic part of the protein. Yeah, and then the peripheral proteins, those were the proteins that were like, in the fluid mosaic model, would be like a protein that's like attached on the periphery. It's like, basically it's attached to an integral protein. And it technically could also be attached on the other side there too. Give me 1 second, I'm getting a phone call. Okay. Anyways, yes, so those peripheral proteins can be here and they can be there. So yeah, let me get my cursor back over here. Make sure I got everything. Yeah, so I want to get into some of the different roles or functions of these membrane proteins. And there's going to be a lot of details in the coming slides. You don't need to know the details. You really just need to kind of have these 6 different functions just memorized. It's not really like you need to sit here and overly worry about, especially signal transduction. That slide's kind of complicated. It's just more of just like the basic definition of these different things. So let me just go over them one at a time. So for A, transport, this one's fairly obvious. Basically, we'll have some molecules that can't cross the cell membrane by themselves, or they can't cross very quickly. They need to have something that's going to help them get across, or get across just in a manner where it's faster, so that we can, the cell needs to have these things quicker. Anyway, so an example would be like an aquaporin. So maybe this is water molecules, where these water molecules can come in through this protein. Basically water would be polar, so it wouldn't normally be able to get through the hydrophobic part of the cell membrane, but they would be able to then just kind of, the inner part of this protein right here, so like the inner part of this channel would be lined with a bunch of polar amino acids that can interact with the polar water. And then the outer portion of this protein is going to be made of the non-polar amino acids because it's interacting with the phospholipid bilayer. So that's the channel. And then there also could be what's called active transport, which you'll learn more about later in chapter 5. Or basically you could have this is called a transporter protein that can transport molecules against their concentration gradient. Anyways, some proteins can act like enzymes. Enzymes speed up chemical reactions. So you could have actual proteins in the cell membrane that will be used to do chemical reactions. When we go over photosynthesis and cellular respiration, you'll see a lot of those proteins be able to do that, where they can, catalyze that chemical reaction. All right, signal transduction. So you'll have some sort of signal molecule. This is where this one can be a little fuzzier for some students. So basically, you'll have some sort of signaling molecule that's going to could be like a hormone, something that's going to, one cell is going to communicate to another. It binds to a receptor, a protein in the cell membrane. That's then going to communicate. So this signal molecule binds to the receptor. It's then going to communicate from the cell membrane to the nucleus. So this signal transduction, this signal is going to be transducted. I think that's a verb. It's gonna send that signal to the nucleus, and then in the nucleus, the cell can then respond to that signal. So basically, there's a whole bunch of proteins that are in between here and then the nucleus. It kind of reminds me like a pinball machine, but that's what's going on. Cell-to-cell recognition, I mentioned this earlier with your glycoproteins. So this is your carbohydrate chain here, where this can be really important with different cells in a tissue layer being able to recognize one another, or in the immune system being able to recognize whether a cell belongs there. Intercellular adjoining, I talked about this the end of chapter 4, where you have those tight junctions, or this could be like the desmosomes, like in the muscle cells, those really tight connections that will prevent the cells from breaking apart. You could have proteins like, this would be like fibronectin would be an example, this protein there. That would be used to connect an integral protein to like a collagen fiber in that extracellular matrix. Okay. Okay, so yeah, I think this is just really just saying what I mentioned with the carbohydrates and cell recognition. Talked about at the very beginning of this video where those carbohydrates can be found. Usually it's on the proteins, but also they could be attached to the lipids. So glycolipids or glycoproteins. And then, sort of how among a species or across different species, how they can be used in recognition is changing the types and lengths of those carbohydrates. Kind of like how you would change maybe the depth or the amount of an arrangement of the ridges on like a fingerprint or something. Okay, I know it's a longer section here, but last bit is this idea of membrane-sidedness. So let me just jump to a picture of it. So basically the idea here is when you make a cell membrane, the inner part of the cell membrane, so they show it in this orangish color. I'll trace it here. This inner part of the cell membrane will actually be made from vesicles. And the outer part of the cell membrane will be made from the lipids that would be inside those vesicles. So I'll color those in red. We're assuming the product that was in those vesicles were lipids, and that's why you'll have membrane-sidedness. That's why like the inner part of the cell membrane comes from the actual membrane from the vesicle that came from the Golgi apparatus, and then the actual outer part of the cell membrane will be something that was an actual product that we made, that was at one point inside that vesicle. So it's sort of a little more of a random topic, but that's what they're getting at here with membrane-sidedness. So asymmetrical, if something's symmetrical, it would mean the same. So because there is a difference between the inner and the outer membrane, that's why we call it asymmetrical. You have those distinct inside and outside faces. Okay, there you go.